Hi everyone, welcome back. So here we're going to start our more specific dialogue, more specific discussion about bacteria, the distinct domain bacteria. So we're going to really explore the anatomy and physiology of their cells, otherwise known as their form, like how they're structured, and also their function, how they work. And we're also going to explore a little bit about their ecology as well, how they interact with other living things, including each other. As we go through this unit, I want you to keep in mind that we're going to see some things that are shared across all groups or many groups, including with animals like us. And so bacteria can be similar in important ways. And they can also be extremely different. We're going to certainly talk about those major differences as we go through this section. But it's definitely a phenomenon that as we learn about other groups of living things, we do tend to learn things more about ourselves. So please approach this with that kind of perspective and a bit of an open mind. We're going to start by talking about bacterial cell membranes. So a cell membrane is something that every cell has. Every cell, by sort of the definition, it has to have this envelope, an outer layer that separates the outside world from the inside of the cell. And for all living things, those cell membranes are composed of phospholipids. Okay, and these phospholipids, they're structured in a very specific way. That is, they have a hydrophilic end, this polar head with a slight negative charge, and then they have a hydrophobic tail that doesn't really have a charge. It's non-polar. So these molecules, when they arrange in a cell membrane like this, they're not glued together. They hold together instead because of the hydrophilic and hydrophobic interactions they have with water. The environment that a cell lives in is mainly aquatic, even like inside of our bodies, for example. And then the inside of a cell is also water, strongly water-based. And so the hydrophilic ends like to point towards and interact with the water, and the hydrophobic ends point inwards into the middle of this phospholipid bilayer. Okay, and that is how these things are structured in virtually all life. You may remember from a past unit that we talked about archaea and how archaea actually kind of deviate from this. They have a phospholipid monolayer. So the outside is the negatively charged polar heads that love water, but the inside, while it is hydrophobic, they're all linked together instead of being separate little hairpin structures. So this is a structure, the phospholipid bilayer, that we see in virtually all life, with the exception of archaea. Now, the phospholipid bilayer isn't just lipids either. It's not composed exclusively of phospholipids. It also has a lot of stuff embedded in it. We're going to see a lot of proteins, different carbohydrates, and other complex lipids, and even like some mixed materials like carbohydrates that are attached to proteins, like a glycoprotein, for example, that are lodged either partially or completely inside of the phospholipid bilayer. And they serve a variety of different functions that we can dive into at a later time, but essentially they're ways that the cell interacts with its environment in different ways. Allows some things to pass freely through the membrane and prevents other things, and so on. Now, a lot of these proteins that are embedded here, they're one of their main jobs is actually moving material, because the cell membrane is what we would call selectively permeable. On its own, without all these proteins in it, this phospholipid bilayer allows some things to freely pass between the phospholipids, to squeeze through the cracks, but not other things. Things like dissolved gases like carbon dioxide and oxygen, they can usually move through these cracks. Also water, like water molecules can squeeze through the spaces between these phospholipids, but charged particles like, for example, ions, like a sodium ion, is not going to be able to move through that space. Also, like larger molecules like glucose and things like that, not going to be able to squeeze between these spaces. And that's where these proteins come in. A lot of them are pathways. A lot of them are pathways that can be open and shut to allow specific things to pass through. Another really important quality of the cell membrane, specifically for prokaryotes like bacteria, is that they have the specific job of making ATP. ATP, this molecule, it's called adenosine triphosphate, ATP. This is, if you're unfamiliar, our main energy molecule, the fuel that we use to power a variety of biochemical processes that a cell has to do. All cells use ATP. Every cell uses ATP in some form or another for powering major biological processes. And so we have to get that ATP from somewhere. So what we do, what all living things really do, is we build ATP partly from the energy that we gather from other resources. So ATP really, it's multiple materials that are squished together, and the chemical bonds that hold them together are somewhat fragile. They're like very, very high energy bonds. And so it takes energy to push these things together. That energy comes from some of these food sources that we often think about, like sugars especially, but also lipids, some proteins and things like that. A lot of different organisms can take these resources and change them, break them down, and release some of their stored energy in order to power biological processes, like pushing together the molecules to make ATP. And then we can take that ATP and use it to power other processes. It's very cyclical. This is what that molecule looks like. So this is, here we have the structure here where we have like 3 phosphates. This is why it's called adenosine triphosphate. And this final phosphate is attached with a bond that, when broken, produces a very exothermic reaction, meaning that it releases a lot of energy. Okay, sometimes people call that a high energy bond. And forming that bond takes a lot of energy input, and releasing that bond also has a lot of energy output. In eukaryotes, like us, this job happens in the mitochondrion. This is why we call the mitochondrion the powerhouse of the cell. Its main job, is making copious amounts of ATP. And so this process actually happens in the membrane of this organelle here, this inner membrane, like along this inner membrane here. But a prokaryote, they don't have this organelle. Okay, so things get really different. We change that up a lot. In a prokaryotic cell, this happens only in the cell membrane because there are no organelles. So there's nothing inside of the cell that's like a little compartment where these processes occur. Instead, the entire cell membrane is lined with the proteins whose job it is to make ATP. Okay? We're going to have a unit that really explores this in more detail, but the way this kind of works is pretty cool. Basically, we have these complex processes that happen in the cell called respiration. It's a respiration pathway. And over the course of that respiration pathway, we make a lot of these molecules that have lots of free hydrogens on them, like this one here, NADH, and this one, FADH2. And essentially what we do is we use these proteins to pull off these hydrogens and shunt them from the inside of the cell to the outside of the cell. And then out here, there's a high concentration of these hydrogen ions. Well, nothing really likes being stuck at high concentration for a long time. So what hydrogen wants to do is diffuse to a place that has a lower concentration. And inside the cell has a lower concentration. But this whole cell membrane acts as a barrier to that. Because remember, charged particles cannot pass very easily through this cell membrane. It can't squeeze through the spaces between these phospholipids. So the cell provides a pathway for it to flow back over the cell membrane. And that happens right here at this little molecule here called ATP synthase. This is a big enzyme, like a complex of enzymes that acts kind of like a water wheel. So if you imagine the hydrogens being like a river, and then this enzyme being like a water wheel, the water in the river flows across the water wheel and pushes it. It forces it to spin. And the force of that spinning can be used to do work. Humans use water wheels to do things like grind wheat into flour, but these cells use this water wheel, ATP synthase, to smush the molecules that make ATP together, basically to make lots and lots and lots of ATP. This is kind of what that looks like. So this is an arrangement of what we call the electron transport chain that is throughout the cell membrane of a prokaryotic cell. So it actually kind of looks like this, where there's just an absolute crap ton of these different enzyme complexes all working together to do this same process and produce copious amounts of ATP. So that is a very important role that the cell membrane does for prokaryotes like bacteria. Beyond the cell membrane, we often see another structure. It's a cell wall. It's a very rigid and protective outer layer that protects the cell and holds it in place. Kind of gives it its shape. So for bacteria, the cell wall is made of something called peptidoglycan. Okay, this is a protein-carbohydrate mix. It's a combination molecule. Now, other prokaryotes like archaea have different kinds of cell walls. There are even some prokaryotes, including bacteria, that don't have cell walls at all. So it isn't a uniform feature. It doesn't occur in every prokaryotic cell, but it is something that occurs in the vast majority of bacterial cells. So peptidoglycan. When we look at peptidoglycan, we have essentially this rows and rows and rows of alternating sugars, a chain of alternating sugars. Okay, and so you don't need to remember the specific name of the sugars, but you should know their abbreviation, NAG and NAM. So these NAG, NAM, they alternate. Through this chain of carbohydrates. And then special proteins link up these different rows of carbohydrates. So they form what we call crosslinks between these proteins, between these chains of carbohydrates that are made of protein. Together, this is kind of like an interlocking chainmail, like an armor that encapsulates the entire cell and gives it both structure and protection. This is another a view of what that looks like here, so remember that they are alternating like this, okay? Yet another view of those specific sugars. So you can see that they're actually pretty different from like a glucose molecule, and they can chain up really easily. We also have some other important features that can be embedded in these cell walls. So it's important to remember also, I'll also mention this, that these sheets of peptidoglycan, right? You can also have multiple sheets of that peptidoglycan that kind of stack on top of each other. It's kind of like wearing multiple layers. So some species have many more layers of peptidoglycan, and some species have far fewer. So among the species that have more layers, you will often see other structures like lipoteichoic acids and teichoic acids. Okay, so these are special proteins. Lipoteichoic acids are anchoring proteins. So their job is to attach the cell wall to the cell membrane so that they don't move freely from one another. And then the teichoic acids, their job is mostly about resource movement. So they're sort of like little portals where materials can travel through them to reach inner spaces within the cell wall. Now, cell walls of different bacteria are not actually uniform. There's actually a lot of variation, as I started to allude to earlier. Some species are going to have very different configurations, and there's two main configurations that we really talk about with cell walls, okay? That is gram-positive and gram-negative varieties, all right? Now, gram-negative bacteria, we'll start there. What we have is we have two membranes, right? We have the inner cell membrane and we have the outer membrane. Technically, this is not a proper cell membrane. It's a bit different, okay? So between them, we have a sandwiched, small, thin layer of peptidoglycan, okay? So they do have a cell wall. It's just very thin. Usually, it's just one to a few layers. Then we have gram-positive bacteria. Here we have this cell membrane, as we usually do, and then outside of that, we have we have a very thick layer of peptidoglycan. Okay, multiple layers stacked on top of each other. Okay, and this gram-positive, this is where we tend to get those teichoic and lipoteichoic acids that really anchor that stuff here and act as like portals that allow things to move through, because the cell wall is so thick that it's hard for some things to move through. Now, for gram-negatives, there's a few more things we want to make sure we talk about, because this outer membrane is pretty important, okay? So, again, on the inside, we have this thin layer of peptidoglycan smushed between two phospholipid bilayers. But the really interesting thing here is that the outer membrane is a bit different in structure compared to the inner cell membrane. We have two major important differences. The first one is that the leaflets are different. So when we talk about leaflets, I want you to think about leaves of paper. Okay, so imagine the outer membrane as two sheets of paper stacked on top of each other. You have one leaflet, the top one, that's the outer leaflet, and then you have the one on the bottom, the leaflet on the bottom, that's the inner leaflet, okay? So this is super interesting because the inner leaflet, while it is composed of phospholipids, the outer leaflet is not. It's actually composed of a different material. It's composed of something that we call lipopolysaccharides, otherwise abbreviated LPS. These are lipids that have special carbohydrates attached to them, okay? And these lipopolysaccharides are really important, particularly in the way that these organisms cause disease, because they can often be toxins in their own right. And so they can trigger the formation of symptoms and also be targets for immune response for cases where they are, they belong to a species that's infecting us. So these gram-negatives do have these additional qualities to them that make them particularly notable. We also have porin proteins that act as pores, right? Pores. Essentially, these are pathways that materials can pass through this otherwise difficult to traverse outer membrane, okay? And so these are very important structures that we only see in gram-negatives. These gram-negatives, okay, there's also another important quality that differentiates them from gram-positives, and that is their resistance to specific antibiotics. Now, a lot of our antibiotics target the bacterial cell wall, okay? These are like penicillins and cephalosporin and stuff like that. they attack the cell wall and do a lot of damage to it, and essentially that's how they kill the cell. They either damage the cell wall outright, or they prevent it from being repaired after it does take damage. And the result of that is that the cells inside the cell wall can actually rupture because they lack now that outer support structure that they were relying on to give them their defined shape. Now, some species are more vulnerable to these types of antibiotics than others. For gram-positives, this is a particularly vulnerable group of organisms for these cell wall targeting antibiotics. And that's because the cell wall faces the outside world. It is the outermost structure for these types of cells. And so there's kind of unfettered access that antibiotics have to the cell wall and can kind of get in there and wreak havoc. Over here for gram-negatives, it's a bit different because that outer membrane complicates things. It's harder for materials to pass through that outer membrane. They can be more selective, and so some antibiotics can't pass that outer membrane very easily. That I want to stress, because this is a common misconception. It does not mean that gram-negatives are flatly resistant to antibiotics, that all antibiotics won't work on them. It means that specific antibiotics won't work on them. So those penicillins, less effective. Some of them may still be effective, but considerably less so, may need to be used in higher concentrations and things like that. We have a variety of different kinds of antibiotics that do a variety of different things. So it's not like just because you have a gram-negative infection that you now can't take antibiotics. Nearly all bacteria fall under this category of gram-positive and gram-negative. There are a few exceptions, but they definitely exist. So the main one is Mycoplasma. This is a group of bacteria that just simply don't have cell walls at all. And so this is actually a pretty interesting and common theme in biology in general, where we have situations where the rules that we have once set forward and believed to be true end up, we find exceptions for those rules. So there's a lot of that stuff. And as you go through biology, as you go through medicine, things like that, you're going to learn a lot of these cases where people once thought all life behaved a certain way, or every organism of this group behaves a specific way or has these features. And then all of a sudden we learn about a rare but notable example of something that defies that understanding. So it's kind of what makes biology so exciting sometimes.